Hi, I'm Jan Witkowski from Cold Spring Harbor Laboratory, and I'd like to welcome you to this, the 88th Cold Spring Harbor Symposium on Quantitative Biology. And the topic this year is brain-body physiology. And I'm very pleased to have Ted Dawson from Johns Hopkins with me to talk about his work on is it synuclein or synuclein? Synuclein, and its role in various disorders. Ted, so tell us, what is alpha-synuclein and how does it come to be involved in diseases?
So alpha-synuclein is this small protein. It's about 15 kilodaltons. It was originally identified as being a presynaptic protein that was enriched in the nucleus. And then in the late 1970s, early 1980s, it was identified as being mutated in Parkinson's disease, an alanine 53 to threonine substitution. And then quickly following that, it was shown to be enriched in Lewy bodies, which is the pathologic defining feature of Parkinson's disease. And then since then, there's just been an explosion of research exploring its function in not only normal physiology, but in Parkinson's disease and related disorders.
So what's the consequence for the protein of that mutation, of that change?
So the mutation makes it increases its propensity to misfold, and it normally has an unstructured, it exists in free form in the cytosol. It's got association to membranes, but what the A53T mutation does is it increases the propensity of the protein to misfold, and it will form oligomers, then protofibrils, and then fibrils, which obtain beta sheet structure. And a number of other mutations have been identified. And the wild type, the non-mutated form, will also misfold. Usually what drives that is oxidative stress or certain types of post-translational modification.
So the normal form of the protein, when you say it too can misfold, that's not because it interacts with the mutant form of the protein. It just that the normal protein can misfold simply by oxidative stress and so on.
Correct. And there's an idea that under normal conditions, alpha-synuclein forms tetramers or higher order structures composed of multimers of the monomer. And for reasons that still aren't fully understood, as long as that is associated to the membrane, synuclein behaves normally, and it's thought to facilitate neurotransmitter release. It's not required, but it facilitates. And then for reasons like oxidative stress, it disassociates from the membrane. The oligomers or tetramer disassociates, and then it leads to a propensity to misfold.
And the misfolded form doesn't associate with the membrane in the way that the normal form does.
Correct.
But what's the what's the consequence of having misfolded synuclein in a cell? The neurotransmission goes awry, or?
Well, so the misfolded synuclein forms these higher order beta sheet structures which can coalesce into fibrils. And these small fibrils disrupt cellular signaling.
Oh, okay.
They can activate cell death mechanisms. We showed that they bind to TSC2 and disrupt the TSC1-2 complex, which can lead to activation of mTOR, which can then set in motion defects in protein synthesis, defects in autophagy, setting in motion this whole cascade of cell death. And then these fibrils, for mechanisms that are still being elucidated, they are released from the neuron that they're formed in and released out, and then they go to the next neuron and are taken up primarily through receptor endocytosis, and then which then starts the whole process over again. And by the misfolded protein templating the normal unfolded alpha-synuclein again to misfold based on the structure of the fibrils that came into that cell.
So this sounds to me very much like a prion-like mechanism.
Yes, and that's one of the major hypotheses now of Parkinson's disease, is that synuclein acts like a prion-like protein where it misfolds, templates the endogenous unfolded synuclein to obtain the structure of the fibril that templated it. And in fact, there's very good biophysical cryo-EM data that synuclein, same protein, can obtain different biophysical and structural properties that seem to be associated with the different clinical syndromes of Parkinson's disease. So patients with dementia have a slightly different form of misfolded synuclein. Fast progressing patients have a slightly different form than the slowly progressing form. Patients with diffuse Lewy body disease have a different form. And then there's another disorder where synuclein misfolds in oligodendrocytes and causes a disorder called multiple system atrophy that also has a different structure. So it's very much like a prion-like disease.
But it's fascinating that it's not fascinating that a variety of mutations can produce varying forms of misfolding. But it's really rather fascinating that the various actually misfolded proteins produce different phenotypes. Right? If I understand what you were just saying.
Yeah.
And if you look at the different misfoldings in relation to the phenotypes they produce, is there any not correlation, but can you deduce anything from a... If I gave you a particular misfolded synuclein, could you tell me what disorder it was likely to produce?
We're not quite there yet, but I think that's where the field is moving. A paper that was published this year or late last year in Lancet Neurology, in which patients with Parkinson's disease who donated their cerebrospinal fluid, they found that misfolded synuclein was in the cerebrospinal fluid. And I could see a day where as these biophysical cryo-EM methods are operationalized, you could go in and get a spinal tap, and based upon the structure of your misfolded synuclein, we will be able to tell you what type of Parkinson disorder you have.
Well, that's a way off, presumably. But your talk, which you're giving tomorrow morning, is on a different, well, on a related topic about spread of synuclein from the gut.
Correct, yes.
So explain that a little bit more.
Yes, so part of this, what led to the spreading hypothesis was work by Heiko Brock in Germany, who did a very detailed pathologic examination of alpha-synuclein expression in patients who had come to autopsy. And he found that patients who had abnormal synuclein in the cortex had it throughout the brain. But then they found patients that it was just in the gut. And then they found patients that it was just in the gut and maybe up to the midbrain. And so he put forward this hypothesis that the synuclein was misfolding, and it started in the gut and ascended up the vagal nerve to cause Parkinson's disease and then the later cognitive symptoms. And what I'm going to talk about is we were able to prove his hypothesis in an animal model by injecting these misfolded synuclein in the duodenum and pylorus of the stomach, and we were able to show, much like Brock proposed, that the synuclein templated the endogenous synuclein, ascended up the vagal nerve to remarkably phenocopy Parkinson's disease in a mouse.
That reminds me of the BSE problems in the UK, where I think John Collins showed that something was but that was, I guess it was somewhat similar, that if you ate contaminated food.
Yeah, you would get bovine spongiform encephalopathy
BSE,
of prion disease.
Yeah.
I certainly would not want to eat the brain of a patient with Parkinson's disease, but I don't think it's as infectious as, classic prion disease. But there are accumulating pathologic studies of patients who receive growth factor, growth hormone extract.
Yes.
That as those patients have come to autopsy, particularly with not regards to synuclein, but with regards to tau and A beta, they're showing that there's probably a reasonable likelihood that they got their Alzheimer's from their growth hormone treatment.
Coming back to what you're saying, so you've demonstrated that if you inject into the duodenum or abnormal synuclein, it can get to the brain. But in the normal course of events, do we eat food that will have abnormal synuclein in it?
No, there is no evidence to suggest that it's through diet. But but this may get at why is the synuclein misfolding in your gut in the first place? And it's probably related to the microbiome. And so there was a study published in Cell probably in 2018 that if you take a transgenic mouse that overexpresses synuclein and gets disease, let's say 12 months later, put it on a germ-free diet, it gets disease not at 12 months, but 18 months. And then if you give it bacteria, a microbiome from patients with Parkinson's disease, it'll now get disease, let's say 6 to 9 months. And if you give it microbiome from normal patients, and then they'll get the disease at about 12. So there's probably what's occurring is there's a shift in the microbiome that is causing the synuclein to misfold, which likely sets the whole motion, the whole cascade in progress.
But that must be a fairly infrequent occurrence, because we all have a microbiome, but I hope my microbiome isn't making misfolded alpha-synuclein and.
Well, it's probably a chicken and egg thing, because patients with Parkinson's disease have an altered microbiome. Is the altered microbiome because of the synuclein causing dysfunction in their GI tract, or did the GI tract create the dysfunction in the synuclein, which is causing Parkinson's disease? I think that's going to be very hard to sort out.
So that's clearly your next your topic for the next time there's a symposium on this topic.
Sure, absolutely.
Thank you very much, Ted.
Thank you.